The meeting is being recorded. Okay. Okay. Hello, hello, hello. We're getting participants coming in from around the world. Good afternoon, everybody. I'm just saying hello as the participants numbers go up. We're kind of excited here in Greensboro because this is the first quasi in-person thesis defense we've done in about 15 months. So we've got technology going, and we've also got people sitting in a room, socially distanced, fully vaccinated, and we're going to put our masks on here in a second. But it's wonderful to have you all coming in. If you want to say what countries you're from, it'd be great. I know that we've got Jordan represented. I'm guessing we might have Mexico represented. Obviously, we have the United States represented. All right. Yes, we see people from Jordan. Hello, Isra. This is kind of interesting because the person who just said Jordan is represented, she was, I think, our first thesis defense totally electronically. And hopefully Zeinab is our last quasi-electronically, but we'll see how that goes. It's probably going to take a little while. Oh, we got the UAE represented. All right, we're going to give everybody just a minute here because we're having a little bit of technical difficulties, and then we'll get started. We have Qatar represented. Hello, Faraz. Wonderful to see you, or sort of see you. What time is it in Qatar? 7 p.m. Okay. 7 p.m. I mean, I have to say, like, there's a lot of things I really don't like about the pandemic, but I have to say it is kind of neat when we have people in many different time zones from all over the world able to watch this. Okay. So it looks like we've about stabilized. All right. Hello, Sherry McFarland. Let me turn something off here. Okay. Well, it looks like we've got people from around the world, including Texas. And, if you're in the United States, you know that Texas is its own country. All right. So it's kind of exciting. As I was saying when we first started here, the committee is actually fully vaccinated and we're all sitting in a room together, socially distanced. And so we've got this presentation going on both electronically and in person. And it's always a little bittersweet when you introduce a student to do their thesis defense because you're very excited for them, but you realize that this is kind of the end of the road for them here in Greensboro, at least temporarily. Every student kind of has their role to play. And any of you who have seen, well, we haven't been to many conferences lately, but if you've ever seen me standing next to Zeinab, you would see that one of my goals in life is to be tall enough to play basketball. And Zeinab makes me feel that way. Right. And the thing I wanted to say about that, hang on, push mute. Got it? Okay, we got feedback going on here. The thing I wanted to say about that is that Zeinab is certainly short of stature. I mean, it's kind of a joke and amongst us, a friendly joke, but her chemistry is very, very tall, very, very tall. If you work in the Oberle's research group, you're going to work on metabolites from nature, probably a fungus, and you're going to isolate some structures, right? I mean, that's what we do, right? But so Zeinab has done that. And in English, we could say she's done that in spades, meaning she's done it over and over again. But the other thing you want a student to do is to kind of move us into new areas and new ideas, occasionally new journals, occasionally new collaborations. And she has done all of those things. And the one thing I wanted to say to the students out there, the students from UNCG, for those of you who don't know this, once a year, we take all of the graduate students and we kind of give them a run through where they have to present their research to the faculty. So one student and all the faculty, and you got about 10 minutes. And interestingly, a very large portion of what Zeinab is going to present today stems from a question that was asked during that session. And I think if you're a student, I try to think back to what it was like to be a student. I'm sure they're a little nervous to stand there in front of the faculty and you're worried like, oh my God, I just want to get through this. I hope they don't ask any questions. But sometimes the questions they ask are really, really good. And so you'll see towards the end of this talk, some questions that Dr. Hamatian asked Zeinab. Dr. Hamatian was probably our brand new faculty person at the time. And you'll see how that really took us into whole new ideas with this research. So, all right, without further ado, we're going to clap from around the world and in person as we let Zeinab take over the presentation. It's all yours, Zeinab.
So thank you, Dr. Belis, for the nice introduction, and hello everyone. As Dr. Belis said, I'm Zeinab Al Subeh, a PhD candidate in his research group, and today I'm doing my dissertation defense that involves the preclinical development of fungal secondary metabolites and the discovery of new metabolites with cytotoxic properties. In other words, in the past 4 years, as well, I was mainly working on the preclinical development of potential anti-cancer compounds, and also I tried to discover new metabolites, fungal secondary metabolites that have cytotoxic or anti-cancer activities. So yes, we're still looking for cancer treatment, and this is because cancer is still the second leading cause of death around the world. So every year there is around 10 million people dying from cancer. And the same situation, unfortunately, is in the United States of America. And the same in my home country, Jordan, where every year there is around 4,500 Jordanians dying from cancer. And among all the different types of cancer that have been identified, we found that lung and breast cancer are the most common cause of cancer death around the world, in the United States of America, and in the Hashemite Kingdom of Jordan. So you all know that human beings have been fighting this disease for many, many years so far. And scientists have made a great progress with discovering and developing many anti-cancer medications to fight this disease. And we were lucky that most of these medications were really coming from nature. So if you look to the anti-cancer medications that have been approved by the FDA since 1981, you can see that only 16% of these drugs were coming just by photosynthesis, while the majority, which is 84%, were either natural products derived from natural product or inspired by natural product or somehow related to natural products. In this slide, I'm showing you just a short list of the primary chemotherapeutic agents that are now being used to fight this disease. And you can see that these different medications fall into different classes because they exhibit different mechanisms of action. And they have been proven to treat different types of cancer, but they also discovered from different origin. So if I color-coded these compounds, you can see that those medications highlighted in green were discovered from plant origin, while those in purple are actually derived from bacterial metabolites. And those highlighted in yellow are actually derived from marine natural products. Surprisingly, none of these medications were coming from fungi, which is weird for us because we all know that fungi was a source for several medications that now being used to treat cardiovascular diseases, antifungal infection, bacterial infection. And in fact, fungal secondary metabolites have saved so many lives, but not cancer patient lives. In our least research group, we believe that fungi are still a potential source for anti-cancer leads. And this is because fungal kingdom that involve between 3 to 5 million species, only 3 to 8% of them have been studied for bioactive secondary metabolite, and much less percent were studied for cytotoxic property. And we all know that fungi have the ability to produce a wide range of chemical diversity of these metabolites. And many of these compounds have exhibited cytotoxic properties. And they exhibit this cytotoxic property through different mechanisms of actions. And the big advantage that fungi provide over other natural sources is that you can easily cultivate and grow these fungi, manipulate them in the lab settings, which is unlike plants, for example, that you need to collect them from their natural habitat. And they're gonna need a long time to grow and develop and give you the compound you're looking for. However, Drug discovery and development from a fungal secondary metabolite has its own challenges too. To be able to develop a drug from fungal secondary metabolites, you need this compound to have a drug-like properties. This is in terms of stability, solubility, physiochemical properties, and pharmacokinetic properties. Also, you need this compound to exhibit sufficient cytotoxic properties against cancer cells and minimal side effect. And luckily, if it's overcome the cancer-resistant issue that we have. The big challenge actually with fungal secondary metabolite is how much supply or quantity you can get from these compounds. To be able to develop a drug from fungal secondary metabolite, you need at the beginning good amount to do chemical characterization and optimization, and then larger amount to do in vitro and in vivo testing, and then a larger amount to go for clinical studies from phase 1 to 3, and then a huge amount to supply the market in case this compound get approved as a drug. And we all know that fungi really provide limited amount of these fungal secondary metabolites, usually in the milligram level. I consider myself a lucky person with having all of these challenges during my PhD on the several projects that I have worked on. And this is what I'm going to talk about today. So my presentation will start with the delivery of eopenfeldin via polymer-coated surgical batteries that we developed to prevent the local reoccurrence of lung cancer. This work was published earlier this year in the Journal of Controlled Release. And then I will show you the media and strain study that we did to enhance the production of cis-enol ricinoleic acid lactone to be used as a feedstock for semesynthesis. This work got accepted last week in the Journal of Antibiotics. And then I will show you the enhanced production and the photoactivated anti-cancer properties of fungal pyridine quinone. And after that, I will shortly go over thiolavins and their tuned biosynthesis before going to the last part to show you how the redox behavior of fungal pyridine quinone can derive their structural diversity in nature. So starting with lung cancer, which as I said earlier, is one of the most common causes of cancer death around the world, treatment of this disease usually starts with surgical resection of the primary tumor. And this is followed by systemic chemotherapy with the goal of eliminating any residual cancer cell that usually stay after the surgery. However, with the systemic chemotherapy, you have a local poor penetration to the lung tissue, which allow for just a small concentration of the chemotherapeutic agent at the tumor site, which is not enough to kill the remaining cancer cells. Also, the concentration of this chemotherapeutic agent will be high in the patient's blood, causing high systemic toxicity. So the overall outcome is high reoccurrence rate, poor prognosis, and low survival rate for lung cancer patients, as we saw earlier. So if we thought about different drug delivery systems to solve this issue, for example, if you have a batteries that's coated with a chemotherapeutic agent, and you implanted this drug-loaded batteries at the tumor site at the time of the surgery. So once you remove that tumor, you can implant this drug-loaded batteries, and the drug will be released slowly over time with a local high concentration that's enough to kill any residual cancer cells. And at the same time, the drug concentration in the patient's blood will be low, so the overall result will be reducing the reoccurrence rate, having a better prognosis, and increasing the survival rate of lung cancer patients. Among all the fungal secondary metabolites that we have worked on in Oberli's lab, we found that eopenfeldin would be the best candidate for such a drug delivery system. This is because eopenfeldin exhibited cytotoxic activity at the nanomolar level against several cancer cell lines. And most importantly, eopenfeldin have a low solubility. In fact, the worst solubility we have ever seen. And this low solubility would prevent the development of this medication as anti-cancer drug, but it's going to be advantage of eopenfeldin will be loaded in such a prolonged release drug delivery system, because the low solubility will allow for a prolonged release rate, and the compound will be released slowly over time. So to make these eopenfeldin loaded batteries, we used PGA batteries made from polyglycolic acid polymer, and we coated this batteries with eopenfeldin mixed with another polymer BCGC18 dissolved in DCM, dichloromethane, and we coated this batteries from each side in a uniform way, and we give it enough time to dry before it's being used. And because at that point, we didn't know what the best design for these eopenfeldin loaded batteries and how to make the release profile slow enough to achieve our goal, we intended to make different formulation of these Eopenfilden-loaded matrices, as you see in this slide. So formulation 6 is just a Planck polymer-loaded matrix that used as a control in the in vivo study, while formulation 1 to 5 were Eopenfilden-loaded matrices, and I will tell you the difference between them. So formulation 1 was loaded with a total of 600 microgram of Eopenfilden from 300 on each side. Formulation 2 was basically the same as 1, except that you have extra layer of Planck polymer on the top of the Eopenfilden layer. Formulation 3 have the double dose of Eopenfilden. We have a total of 1200 microgram of Eopenfilden, 600 on each side. 4 and 5 are basically the same as 3, except that formulation 4 have extra layer of Planck polymer on the top of the Eopenfilden layer. And 5 have 2 layers of Planck polymer loaded on the top of the Eopenfilden layer on each side of the matrix. And our goal of having these Planck polymer layers was to see if the Planck polymer will act like a physical barrier that will help slowing the release rate of the Eopenfilden from these Eopenfilden-loaded matrices. And the next step was to study how the release profile will be for these different formulations to decide which one is the best to go with. So for each Eopenfilden-loaded matrix, we placed each one in 50 ml phosphate buffer saline with a small percent of surfactant. And we followed the system for 3 months, where at certain time points, we were taking aliquot from the buffer samples to quantify how much Eopenfilden gets released. And at each time we're taking aliquot, we were replacing the whole buffer with a fresh one to make sure that the system is not getting saturated. And we developed a calibration curve for the Eopenfilden that was used to quantify the amount of Eopenfilden that gets released from these buffer samples. And we get these nice data. So in figure 1, which represents the cumulative release profile of Eopenfilden, you can clearly see that formulation 1 and 2 had a fast release rate from the beginning, where you reach the plateau stage at day 20, which means that the whole Eopenfilden amount from these two formulations gets released on day 20. And after that, there is no more Eopenfilden. And this is not achieving our goal, because our goal was to have a prolonged release profile that can reach approximately 3 months. Formulation 3 had relatively slower release rate, but we also reached the plateau stage at 35 days. And 4, because it has extra layer of Planck polymer, compared to 3, it has a slower release rate, and we reached the plateau stage later at day 55. But luckily, that formulation 5, it was the one that's showing the slowest release rate compared to the other formulation. And it's the one that was really successfully achieving a prolonged release profile that approximately reached 3 months. Figure B is kind of the same as A. We just represent the data as a percentage. And figure C showing you the release per day of Eopenfilden. And you have almost the same conclusion. You can see that formulation 5 was the one that gave us the smallest release amount of Eopenfilden over time. And it was the only formulation that able to keep significant amount of Eopenfilden getting released at the end of the study until 3 months. Figure D is showing a result of extra experiment that we did to make sure that our measurement is actually accurate and precise. And for this experiment, what we exactly did is extracting the Eopenfilden-loaded matrices from the 5 formulation at different time points. We did it at time 0 before exposure to buffer, after 1 month, 2 months, and 3 months. So you can see that at time 0, we were able to get significant amount of Eopenfilden, which relatively represent the amount that we loaded at the beginning on this formulation. At day 30, we had no Eopenfilden extracted from formulation 1 and 2, which matched the release profile that we just saw, where these two formulations release all the Eopenfilden by day 20. So by 30 days, you will not get anything remaining. Only formulation 3, 4, and 5 had significant amount of Eopenfilden remained on the matrices, with formulation 5 has the highest significant amount. And after 60 days, only formulation 5 had significant amount of Eopenfilden remained on these matrices, around 50 microgram, and this amount gets released in the next month, where by day 90 you have no remained Eopenfilden. So this study tells us that design 5 for Eupenfilden loaded matrices is the best one to give you a prolonged release profile. And the next step for us is to see if this prolonged release profile will translate well into prolonged cytotoxic activity on in vitro using lung cancer cell lines. So we tested these 5 formulations against lung cancer cell lines, the LLC cell line and the A549 cell lines. And you can see clearly that there was a potent cytotoxic activity for the 5th formulation at the beginning until week 4. But only 3, 4, and 5 were having prolonged cytotoxic activity that stayed until week 10, with the trend of having formulation 5 being the most potent one compared to 3 and 4. Against cell lines A549, it's kind of the same conclusion or the same trend in the data, although Eupenfilden looked like to be less cytotoxic against these cell line, but still formulation 5 was the one that showed the most potent cytotoxic property with prolonged activity reached 7 weeks. So again, this study also confirmed the same conclusion that formulation 5 is the best design to give us a prolonged release profile. So we choose this one to go for the in vivo study that was conducted by our collaborator at Harvard Medical School. And they used animal model using mice. They injected these animals with LLC cell lines, and they waited for the tumor to get into a good size before doing the surgery to resect the tumor and at the same time implant the Eupenfilden loaded mattress. And then the animal were followed to see how the survival and the reoccurrence rate will be. And we get these nice data. You can see clearly that the group treated with Eupenfilden loaded mattresses were having significantly reduced reoccurrence rate compared to the animals treated with blank films all control. And also we have the overall survival and the disease-specific survival were significantly improved in the treatment group compared to the control or the group treated with a blank mattress. So this is really nice to see how the efficacy of this Eupenfilden loaded mattress was effective in reducing the reoccurrence rate of lung cancer with these animals. And in the next slide, I'm showing you how the animals treated with Eupenfilden loaded mattresses didn't show any signs of systemic toxicity in terms of cardiac or hepatic toxicity as compared to the healthy control. And this tells us that the Eupenfilden loaded mattresses were effective in achieving higher efficacy and at the same time reducing the systemic toxicity. And this is the main 2 goals of this study that we were able to successfully achieve. And with this, we ending the story of Eupenfilden loaded mattress and their role in preventing the local reoccurrence of lung cancer. And next, I will be talking about the media and strain study for the scaled production of cis-enol resulfuric acid lactone to be used as a feedstock for synthesis. So as the name indicates, cis-enol resulfuric acid lactone are a group of fungal secondary metabolites that have the cis-enol moiety. You have a double bond next to a carbonyl. And hypothyamycin and 5Z7-oxazienol shown in this slide are the 2 main compounds belong to this class. And this class gained a significant attention by scientists because of their role as an irreversible inhibitor of kinase enzymes. So they react with the thiol group in the cysteine amino acid in the kinase enzyme to make a covalent double bond, and this will cause irreversible inhibition of these enzymes. And we all know that kinase enzymes play a major role in cancer growth, development, and aggressiveness. So this irreversible inhibition of these enzymes will translate to a potent cytotoxic activity. And Dr. Croat and Dr. Uberlis have a grant to further characterize these compounds and push them as anti-cancer lead. However, as I said before, to do this study, you need significant quantity of these natural products, hypothyamycin and oxazienol, and this was my job to do. So to achieve this goal, we screened around 530 fungal strains from mycosynthetic company against our dereplication database. And we were able to identify 7 fungal strains as a producer of resulfuric acid lactone. Of these 7, 3 of them were a producer of either hypothyamycin or oxazienol or both of them. And from the experience we get in Uberlis research group, the first smartest thing to do to enhance the production of any natural product is to study the media that will enhance the biosynthesis of these compounds. So for each one of these strains, we grow them in 3 different media. We use rice, oatmeal, and Cheerios, and we grow them in triplicate. And then we did, after the incubation, we did extraction, and we subject the extracts to UPLC mass spec system, where the data were used to measure the relative abundance of the compounds that we're looking for, hypothymycin and oxazionol. For hypothymycin, you can see that we were able to detect hypothymycin in the extract of the 3 strain, but it's clear that the relative abundance was significantly higher from the extract of strain 78495. With relatively speaking, oatmeal media give us higher abundance compared to rice or Cheerios. So from this data, we know that if we need more hypothymycin, we have to grow strain 78495 and better to use oatmeal. For oxazionol, this compound was detected in the extract of 2 strains, 78495 and 63935. And it's very clear that oxazionol abundance was more in the rice media as compared to the oatmeal. And it's interesting to see how the Cheerios kind of significantly suppress the biosynthesis of this compound. And this tells us the importance of the media and the media study to do if you're targeting specific natural product. So for oxazionol, now we know that if we need more of this compound, we have to grow strain 63935, and it's better for us to use the rice. The next challenge that we have with these 2 compounds is how we're gonna improve the isolation and the purification process of them. In Oberle's research group, we provide many fungal secondary metabolites, and the traditional technique that we use is having a culture do extraction, and then we do flash chromatography, and then we mainly depend on the HPLC system to get a pure metabolites. And this is really efficient technique if you're targeting 1 to 100 milligram of a compound, but if you're looking to a gram scale, this is not really the case. So if you want to purify a gram of hypothymycin, you're gonna have the culture, you're gonna do the extraction, go to flash chromatography, and then on the HPLC, you will need to do around 15 injections on the HPLC prep system. And those who worked on the HPLC will know that 15 injections will give you around 10 to 12 liters of solvent between aqueous and organic. And how much hard to dry down this fraction using the rotabab. So in approximately, you're gonna need 2 to 3 weeks of continuous work to get 1 gram of hypothymycin, and another 2 to 3 weeks of work to get a gram of oxazionol. So the other challenge with the purification of these compounds, that even with using the HPLC, you can look to the chromatogram how the hypothymycin peak in here is overlapping with the next peak, and oxazionol also peak overlapping with the next peak. And this is why you can't inject more than 50 to 100 meg of fraction on the HPLC per injection. And even with the 50, you get this overlap in the chromatogram. This picture is showing you the oxazionol, how it come out from the HPLC in the tube. It's crushed out of solution immediately. And this tells you how bad the solubility of this compound is and how hard to deal with these compounds. So to improve this isolation and purification technique, our thought were going over the optimization of the flash chromatography stage. So we get the culture from AMA67845, we did the extraction, and we tried to improve the method on the flash chromatography. And we get a fraction of 640 meg pure hypothymycin, 97% pure. But we also get another fraction around 650 that mainly have hypothymycin, but not pure enough. So instead of going to the HPLC, we found another alternative, which we know a technique called resuspension centrifugation process. And the idea of this technique is to resuspend the fraction in a small volume of organic solvent that have low solubility, that hypothymycin have low solubility in. So we resuspend this fraction in 12 ml methanol, and we vortexed and we did centrifugation to collect the precipitate, and we did another cycle of resuspension and centrifugation. And luckily, we end up having a precipitate of pure hypothymycin, 94% pure hypothymycin, and the quantity was 0.5 gram. So instead of spending weeks on getting 0.5 gram of hypothymycin, this method will take 1 hour or 2 hours in the lab, and you get good quantity of this compound. Then we wanted to see if this technique will work also for oxazionol. So for extract from AMA66935, we did the extraction, we did the flash chromatography, and then we get a fraction around a gram of oxazienol containing fraction that's not really pure. And we did resuspension the same as we did with hypothymycin. We did resuspension centrifugation 2-cycle in methanol, but the precipitate wasn't pure enough. So we did another 2-cycle of resuspension centrifugation, but now with using different solvent, which is acetonitrile, and we end up having 750 milligram of 94% pure oxazienol. So this tells us that the resuspension centrifugation technique is really efficient method to remove all the impurities and other secondary metabolite that we don't need and end up having a precipitate of the targeting compound. So then we processed the supernatant from the 2 strains to see what kind of compounds are getting there. So we purified these fraction on the HPLC. From the MS67845, which is the hypothymycin producer, we were able to identify 6 compounds. One of them is hypothymycin. The other were analogs in the same class, risosolic acid lactone. And from the supernatant collected from MS63935, which is the oxazienol producer, we were able to isolate larger group of compounds. Actually, they fall into 3 different classes. We had 8 in the risosolic acid lactone family. We have 6 in the benzopyrenone class. 2 of them were really new. We described them to the first time of the literature, and 2 were in the redicinal and dihydroredicinal. So this tells us that the resuspension centrifugation technique is really efficient in removing all the fungal secondary metabolite that we don't need, regardless if they are structurally similar to hypothymycin and oxazienol or from different classes. And using this media and the strain study, it's really helped us to scale up the quantities that we have from oxazienol and hypothymycin and make Dr. Croat and Dr. Uberlis dream true. So for now, we have around 70 grams of hypothymycin and 3.6 grams of oxazienol. And it's really worth mentioning that these compounds are really priceless. So if you want just to buy 1 milligram of hypothymycin, you need to pay $570. And it's gonna cost you $180 to get 1 milligram, just 1 milligram of oxazienol. So the quantity that we isolated, it's really worth more than $4 million. Just to make it clear, I didn't get any of this money. I'm still a poor PhD student. But me and my undergrad, Jennifer, who really helped me scaling up these compounds, had the chance to get this nice picture with these 2 great scientists, Dr. Uberlis and Dr. Croat. And this is for us worth more than $4 million. And with this, I'm ending the story of risosolic acid lactone, and now I will be talking about how we enhance the protection and the anti-cancer property of photoactivated pyrrolinequinone, a work that got published last year in the Journal of Natural Products. So pyrrolinequinone are fungal secondary metabolites, and under this family, there is a subclass called hypocrylene. And hypocrylene, mainly involved in the literature, there is 4 compounds in this class. These are hypocrylene A and hypocrylene, which is the enantiomer of hypocrylene A. There is shirachrome A and hypocrylene B. As you see, these compounds have really unique structure with having a highly conjugated pentacyclic core. And they also have something we call axial chirality. And what I mean by axial chirality, that the aromatic rings are not really planar. They are twisted out of planarity in one of 2 directions. These compounds gain really attention by scientists because they act as natural photosensitizers. And by photosensitizers, I mean that these compounds, when they're exposed to light, they tend to react with the oxygen molecule to give reactive oxygen species and free radicals. And these free radical and reactive oxygen species will give you potent cytotoxic activity against cancer cell lines. Also, it's going to exhibit antibacterial, antifungal, and even antiviral activity. Of course, in Uberlis research group, we are more interested with their anti-cancer properties. And we were lucky to cross over one of the fungal strain from mycosynthetic, which is MS66059, that's producing these kind of compounds. And then to be specific, it's producing hypocrylene, and a second compound that was new also, we identified as the enantiomer of shirachrome A. To study these compounds, we need again more amounts, and unfortunately, the first amount we isolated was 2 to 3 mg of each compound, which is clearly not enough to work with. So we had to scale up these compounds. And as I said before, the first smart thing is to study the media. So we did the same thing as we did with ricinoxalic acid lactone. We grew this fungus on 3 different media: rice, Cheerios, and oatmeal. But we also introduced another factor to this study, which is the light exposure. And this is because these compounds, as I said, they are photosensitizers. They get activated with light exposure. So we were interested to see if we expose the culture to light, would this enhance the biosynthesis of these compounds, or it would suppress the biosynthesis of these compounds. So from each media, we put some culture growing under light-dark shift, 12 hours under light, 12 hours under darkness, some culture growing under continuous LED light exposure, and some under complete darkness. And here I'm showing you the LED box that we use to grow this culture under LED light. You can really choose what wavelength and color you need, but for this study, we just use the white LED light. And this is after 2 incubation, the picture of the cultures. And we did the extraction, and we subject the extract to UPLC mass spec system. And we use this data to measure the relative abundance of the targeted compound, which is Shiraya A enantiomer and hypochrylin. And here you can see clearly that Shiraya A enantiomer had the highest abundance on rice media. And to be specific, the rice media that's placed under light-dark shift or continuous LED light exposure. But those under darkness have significantly suppressed biosynthesis of this compound. And the same trend was shown with hypochrylin. And with the actual isolation of these 2 compounds from the different extract, you can see that we were able to get the highest amount of Shiraya A from the same rice media that we identified with the relative abundance. We get around 100 meg from each condition. And for hypochrylin, we get relatively lower amount, between 7 to 10 meg of these 2 conditions. So now we know that to get more of the hypochrylin compound, Shiraya A enantiomer or hypochrylin, we need to grow the MSX 60519 on rice media, and definitely we need light source. The second interesting thing that we found through the analysis of the UPLC mass spec data is the 3 peaks that we saw in the extract of culture grown on oatmeal and under LED light exposure. These peaks were showing UV profile that's similar to that observed for hypochrylin, but we didn't know what these compounds are. So we processed this extract and we isolated these compounds to get a second group, which is called hypomycins. We get hypomycin A, C, and the new compound hypomycin E. So as you see, these compounds are structurally similar to hypochrylin class. The only difference is that the conjugated system is kind of disturbed with losing the double bond between position 1 and 2 in the 3 compounds. We have also extra 6-membered ring cycle next to the pentacyclic core. So we went back to the extract to measure the relative abundance of these hypomycins compound among the different conditions. And it's nice to see that they all follow the same trend, where the highest abundance was observed for culture grown on oatmeal media, and specifically the one that placed under continuous LED light exposure. So now we know that if you need the second class hypomycins of these compounds, you grow the same fungus, but now you need to use oatmeal, and you definitely need LED light exposure. So just to talk, so now we have 6 compounds. We have 3 in the hypochrylin family and 3 in the hypomycins. And you will see that I added a new compound. It's not a new, it's known, hypochrylin B, but a new to the list. Hypochrylin B is known as fungal secondary metabolites, but I was able to make it by semi-synthesis, just simple dehydration reaction from Shiraya A. And we sent these the 6 compound to our collaborator, Dr. Sherry McFarland, who really helped us to test the cytotoxic properties of these compounds with light exposure against melanoma cancer cell lines. And you can see that the 3 compounds in the hypochrylin family showed potent cytotoxic activity with light exposure, either the blue or the red light, and the activity was in the nanomolar level. Hypomycins A, C, and E were really less light sensitive and less cytotoxic with light exposure, with cytotoxicity at the micromolar level, and it was more potent with the blue light exposure than the red light exposure. So just to talk briefly about the structure elucidation of these compounds, as I said earlier, these compounds have something we call axial chirality, where the aromatic ring are twisted out of planarity in one of two directions. And you can identify the axial chirality of these compounds by measuring the ECD spectra for them. So we measured the ECD spectra for these compounds, and we were able to successfully identify the axial chirality for these compounds, specifically for hypomycins, because these were never studied in the literature before, and hypomycin A and C, although were reported before, they were reported as relative configuration, but not the absolute. And to suggest the absolute configuration of hypomycin's compound, we did more work on hypomycin A, where we used the ECD calculation to confirm the axial chirality of hypomycin A. And we used VCD calculation to confirm the absolute configuration of hypomycin A, where it showed 65% similarity with the structure marked on red compared to 11% similarity to the other suggested structure. So we suggested the absolute configuration of hypomycin A in this study. And just by comparison, we didn't do any calculation for hypomycin C and E, but we assumed that they're gonna be similar to hypomycin A, and we used NOSI data to figure out the difference. And by doing this, we try to suggest the absolute configuration of these two compounds. In this slide, I'm showing you a third class of compound called thilavins, which I was able to isolate from the same fungus, MS660-519, that give us hypochlorine and hypomycins. These four compounds, when we look to their relative abundance among the different extracts, you can see that the highest abundance was on rice media, and specifically under light-dark shift condition, where LED light exposure kind of significantly suppressed the biosynthesis of these compounds. So it's really nice to see that there is three classes of compounds produced under three different conditions, and you can manipulate the culture to make it more of the compound you need. So if you need hypochrylins, you know that you should grow on rice and you have light-dark shift or LED light exposure. If you need hypomycin, you should use oatmeal media and LED light exposure. If you need thilavins, you use the rice and light-dark shift. So we published this work on the hypochrylins and hypomycins in the Journal of Natural Products, and it was really interesting data, but it also raised more questions to us. And one of the questions that we had is, if these compounds are really photosensitizers and they exhibit toxicity with light exposure, then how exposing the culture to light significantly increase the biosynthesis of these compounds without affecting the growth of the culture, how the fungi, in other words, protect itself against these toxins. And the other question that we had is about hypomycins. Why these compounds are showing up under a specific condition? You saw that they are structurally similar to hypochrylins. So is the fungus producing hypomycin from hypochrylins under certain conditions? We looked to the literature. There was no study on this class of compound, but there is evidence from a structurally similar compound called cercosporin, which is a photosensitizer. And they suggest that the fungi producing this photosensitizer keep it in the reduced form rather than the oxidized form. And the reduced form is less sensitive to light, so it's less toxic. And this is how the fungus protects itself from this compound. So we needed to see if the theory will apply for hypochrylins and hypomycins. And to do that, we move to the next topic, which is the redox behavior of pyrrolinoquinone and how it's going to drive their structural diversity in nature. So, as I said, we were interested to study the redox behavior of hypochrylins and hypomycin, but before doing that, we needed more quantity of these compounds. So we scaled up the culture to get more of these 6 compounds, and we were lucky to be able to isolate a new compound that's similar to the hypochrylins, and we were able to solve the structure by mass spec and NMR data. And the interesting thing about this compound is its high similarity to the synthetic precursor used in the total synthesis of hypochrylin A. And this made us think that maybe this compound is really the biosynthetic precursor of the hypocrylins family, where the fungus make this parent compound first and then convert this compound to Schreier-Chrome A enantiomer or hypocrylin. And we looked more in the literature. We found many papers talking about the biosynthesis of pyrroliniquinone suggest this structure as the parent compound for several classes in the pyrroliquinone family. So if you have this compound and you did reduction of the ketone side chain, you end up having a class of fungal secondary metabolites called calfostine and cercosporine. If you did oxidative enol coupling, you have another fungal secondary metabolites called encinochromes. If you get aldol cyclization, you have hypocrylins. So it was interesting to be able to be the first people to isolate this compound and characterize it and suggest it as the parent compound of these classes. So then we studied the redox behavior of hypocrylin compounds and hypomycin compounds using the cyclic voltammetry. And you can see how the CV for these compounds having the typical duck shape, which means that the reduction process is fully reversible for these hypocrylins and hypomycins. And based on the reduction potential, the reduction process is easier or more reversible for the hypocrylins than hypomycins. And then we studied the UV profile or the UV spectra of N-Schreier-Chrome A in the oxidized and in the reduced form. So in this figure, you see that the spectra in red is referred to the N-Schreier-Chrome A in the oxidized form, the quinone form. And you see that we have lambda max at 430 nanometer, while the one reduction with the semiquinone form, with the semiquinone form, the UV spectra is totally different, and now it's expanding to reach the 900 nanometer, and you have lambda max at 620 nanometer, while the fully reduced form, the hydroquinone form, have lambda max around 430, and it's clear that the fully reduced form has less light absorbance compared to the oxidized form, and this go with the theory that this compound is less light sensitive and less photosensitizer compared to the oxidized form. So yes, maybe the fungi is keeping it in the reduced form to protect itself from the toxicity of this compound. But at that point, we didn't have any evidence if the compound really make this compound in the reduced form. So next we were interested to see the chemical reduction of these compounds. What's going to happen? So for hypocrylin, Schreier-Chrome A enantiomer, hypocrylin, and hypocrylin B, we did a small scale reduction reaction using sodium dipynite as a reducing agent. And you see that once you add the reducing agent, the color change completely from red, deep red to yellow. So the reduced form of this compound is really yellow. And because this reaction was made under room conditions, there is regular oxygen supply. You can see that on the surface of each reaction, the compound go back to the oxidized form because there is oxygen around. So this goes with the data we get from the CV, where the reduction process is fully reversible. Once you add a reducing agent with exposure to oxygen, it go back to the oxidized form. And we subject this reaction to the mass spec system to see what we have, and we were able to confirm that we recovered the compound in the reduced form. And we also get another side product in this reaction with masses referred to replacement of one or two of the methoxy group with a hydroxy. And the same observation was for hypocrylin and the same for hypocrylin B. To characterize this product, we scaled up this reaction using N-Schreier-Chrome A, and we were able to isolate the three compounds, compound 8, 9, and 10, where in 8, you can see that the methoxy group at position 11 gets replaced with a hydroxy, and the same with 9, but with the methoxy group at position 2, and compound 10, both methoxy at position 11 and 2 get replaced with a hydroxy. So this is the result of the reduction reaction under aerobic condition. There was regular oxygen supply, and we were interested to see if this reaction, how it's going to perform if we have it in oxygen-free environment, anaerobic conditions. So we did the reaction with N-Schreier-Chrome A and the oxygen-free environment, and we get totally different side products. And these side products, we were able to identify them as hypomycin E, hypomycin C, hypomycin A, hypocrylin, and a little bit of hypocrylin B, in addition to 2 or 3 compounds with the masses correlate with the loss of 1 or 2 of the methoxy. So these data were really exciting because all of these products are really the natural fungal secondary metabolites that we isolated from the fungus. So indirectly, this indicates that the fungus was able to reduce hypocrylins under oxygen-free environment to give you this derivatives, hypomycins and hypocrylins. So we scaled up this reaction under anaerobic condition, and we were able to isolate all of these side products. And then we wanted to explain how N-Shiraya Chrome A structure went to all of these derivatives. So we started with how N-Shiraya Chrome A give us hypocrylin. And this is interesting behavior because to go from N-Shiraya Chrome A to hypocrylin, you need to twist the axial chirality, and you need to change the stereochemistry of the acetyl group. So how that happened? Our theory was that this structure in N-Shiraya Chrome A, you see the acetyl group attached to position 15. This acetyl group is kept in the ketone form because of the hydrogen bonding with the hydroxy group. And this hydrogen bonding is what keeps this compound in the ketone form. But if you disturb this hydrogen bonding, what's going to happen is that N-Shiraya Chrome A will go to the enol form, where the double bond will be stabilized by the conjugation with the pentacyclic core. And if this enol form will go back to the ketone form, it will either give you N-Shiraya Chrome A or will give you hypocrylin. And this is the explanation that we have. And to prove this theory, we disturb the hydrogen bonding in N-Shiraya Chrome A by dissolve it in DMSO, which is known to be a strong hydrogen bond acceptor. And in this slide, I'm showing you the first one is the proton NMR of pure sample of N-Shiraya Chrome A before exposure to DMSO. But after exposure to DMSO, you can see that there is a new set of peaks showing up, and these peaks correlate with the proton NMR of hypocrylin. So you really don't need an aerobic reduction reaction to make N-Shiraya Chrome A give you hypocrylin. You just need to disturb the hydrogen bonding with DMSO, and then you force N-Shiraya Chrome A to give you hypocrylin compound. The next thing is how N-Shiraya Chrome A give you hypocrylin B. I already mentioned that hypocrylin B is just a dehydrated product of N-Shiraya Chrome A, and you can actually get this compound from many compounds in the hypocrylin family. So dehydration reaction of any of these compounds, N-Shiraya Chrome A, Shiraya Chrome A, hypocrylin A, or hypocrylin will give you hypocrylin B. And as you see, hypocrylin B, I'm not drawing any axial chirality for this compound because we didn't see any ECD spectra for this compound. And this was explained to, in theory, that this structure is either planar or it's a racemic mixture of the two enantiomers. And we will be the first people to report the crystal structure of hypocrylin B that confirm that it exists in a racemic mixture of the two enantiomers with the two axial chirality, and that's why you don't see ECD activity for this compound. Then the next question, how N-Shiraya Chrome A give us the hypomycins, hypomycin A, C, and D. And the first observation that we have is the yield of hypomycin A, which is 0.5%. compared to 11% and 5% of hypomycin E and C. And if you remember from the culture, the highest yield that we get was for hypomycin A. So why now the reaction that we did give us low amount of hypomycin A? To explain that, our theory that hypomycin A is not really coming from N-Shiraya Chrome A, but what's going on that during this reaction, some quantity of N-Shiraya Chrome A get converted to hypocrylin, and hypocrylin underwent through the reduction process to give you hypomycin A. And what supports this theory is the agreement or the match in the axial chirality between hypocrylin and hypomycin A. But to test this theory, we did anaerobic reduction of hypocrylin, and we end up having a high yield of hypomycin A. We get 65% yield of hypomycin A. And this study overall tells us that now we know that hypomycin are really derived from hypocrylin class, and we know for sure that hypomycin A coming from hypocrylin, while hypomycin C and E are derived from N-Shiraya Chrome A. And this data really helped us to revise the structure of hypomycin A, C, and E. So now, because we know from where these compounds come from, we looked back to their original compound, and you can see that hypomycin A and hypocrylin are in good agreement in terms of axial chirality and stereochemistry. But the previously suggested stereochemistry for hypomycin C and E wasn't really matching with one chiral center. So it's R in N-chirachrome A, but we suggested to be S in C and E. So now we know that it should be R, that as in N-chirachrome A, that we had a cluster 4 to confirm. And this helped us to revise the structure of hypomycin C. And we know that we should match it with N-chirachrome A, not hypomycin A. And with this, I'm ending my presentation today. And in summary, I showed you the delivery of eopenfilden via polymer-coated surgical patches and how we successfully be able to prevent the local reoccurrence of lung cancer. We also showed how the media and strain study helped us scaling up the cis-enol resolubilic acid lactone. We talked about the photoactivated pyrrolinoquinones and briefly on thiolevins. And we will end up seeing how the redox behavior of pyrrolinoquinone derives their structural diversity. And before going to your questions, please allow me to thank my advisor, Dr. Nicholas Uberlis. Dr. Uberlis, it was really been a great honor to be your student. Thank you so much for everything you taught me and for looking after me. You made me a good chemist and a better scientist. There's really no words that can describe how much grateful I am and how lucky I consider myself to be your student. And I really wish that I met your expectation, at least the minimum expectation from short Jordanian students. I would love also to thank my committee members, and I consider myself lucky to have a chance to work with them closely on several projects and learn from such a great scientist. Thank you, Dr. Chek, Dr. Colby, Dr. Himatian for being here today. Dr. Croat, thank you so much. Thank you for all the people who trained me in Uberlis research group and helped me mastering all the isolation, purification, structure elucidation technique. Thank you, Dr. Tamam, Dr. Pepe, Dr. Muzaifa, Dr. Laura, and Mr. Tyler. Thank you for all the colleagues and the friends in Uberlis research group and in the chemistry and biochemistry department. You were really my big family in Greensboro. Special thank you for my close friends, Herma, Sumaya, Laura, and Diana. Thank you for all the researchers, scientists, professors, PhD students who I had the chance to work with and learn from US and outside. Thank you for Jennifer for helping me a lot in the last year. And special thank you to my lovely school, Jordan University of Science and Technology, for supporting me with a full scholarship to finish my PhD at UNCG. And thank you for all the people who are joining us today from countries around the world, from Jordan, Egypt, Qatar, Emirates, UK, maybe Mexico, and several states around the US. Yes, and finally, I would like to thank my family, especially my parents, my mom and my dad, for believing in me and for their prayers over the day and the night. I love you so much and I missed you a lot. Hopefully we'll meet you soon. Please allow me to say that in Arabic so they can understand me. بالنهاية بحب أشكر عائلتي بالأخص والدي ووالدتي. شكرا كثير على تعبكم وعلى صلواتكم ودعواتكم بالليل والنهار. إن شاء الله يكون مصدر فخر لكم. بحبكم كثير واشتقت لكم أكثر. ربنا يجمعنا على خير إن شاء الله. And with saying this, thank you all again, and I think I'm ready for your questions.
Okay, that was great. I even understood her Arabic. At the very end there, she said, you know, inshallah. And I always joked with her, for those of you from outside of the United States, there's a city near us called Charlotte. And if you say inshallah really fast, it sounds like in Charlotte. And I would always say, no, we're not going to do that in Charlotte. We're going to do it in Greensboro. So that was our joke. But basically, inshallah means God willing. So thank you, Zeinab. Okay, so we have questions that are, so because of a little bit of technical difficulties, I'm going to read the questions from the chat so that Zeinab can hear them. She couldn't hear them. We might be able to get somebody to come and ask a question as well. The first question came from Rufaida, one of our former students. She said it was a great talk, full of hope and enthusiasm, but I still find fungi ugly, in all caps. Well, we'll forgive her for that. But anyway, she wanted to know, have you tried derivatizing secondary metabolites to ease their separation? Yeah, of course she would ask that question. Yeah, yeah. Oh, you know what? You're on mute. You're on mute. I'm sorry.
Okay. So thank you, Rafida, for the question. Yes, we did some derivatization for some natural products, but not for the goal for making their isolation easier, because I believe it's going to make it harder. So it's easier always to isolate and purify the natural product, and then you do the derivatization, because otherwise you will end up having different derivatives from any reaction you do, and this will make the isolation process harder. At least this is from my point of view.
Okay, so we're going to have to do like a mute unmute here thing. So let me ask a second question. This comes from Dr. Chu. He says outstanding achievements. He has two questions. He wants to, the first question is, well, I'll just ask it. You can answer this question first. He's talking about the buttress. He wants to know, does the buttress have to be removed after the treatment? Hold on, let's see.
Answer that. Thank you for the question, Dr. Chu. The answer is no, we don't need to remove the buttress after implanting the buttress, because as I said, they are made from polyglycolic acid polymer, and this polymer is biodegradable and it's safe. So it's just gonna biodegrade by itself over time, and you don't need to take it back.
Perfect. Okay, and his second question was, with regards to using the centrifugation to purify your two compounds, did you try to repeat the centrifugation steps to further improve? So he wanted to know, could you take it even farther, basically?
So as you saw, Dr. Chu, we did kind of 2 cycles of resuspension centrifugation for hypomycins, and we did kind of 4 for oxygenol. Yes, you always can do more cycles, but there is a limit that you should stop there, which is when you the supernatants have your target compound more than the impurities, and then at this stage it's not really efficient. And I can show you a slide that I have here. Let me. So this slide show you the proton NMR of the different supernatants that we collected from each cycle as compared to the precipitate. So you see how the supernatants proton NMR look ugly for the first cycle. It gets a little bit cleaner for the second, cleaner for the third and the fourth, and the precipitate is kind of nice, and the cleaner proton NMR correlate with oxygenol structure. So when you reach the stage where you have far more oxygenol in the supernatant than the impurities, I believe you should stop there. So it's kind of balanced between how much you're going to lose compared with the impurity.
Okay. All right. So now we have another question. This one comes to us from Georgia. This is from Dr. Kevin Liu. He says, great work. My question is a naive one. How will you translate your valuable agents into cancer therapy and money?
So translate our valuable compounds is going to be, I think, with the help of pharmacologists like Dr. Kevin Liu, who will help us doing more in vitro and in vivo study and prove that this compound could really be a potential anti-cancer leads. And I don't really care about the money that much as a scientist, because our goal is to find a good treatment to fight this disease. So, yeah.
Okay. Now we have another question. This is from Mohammed. What a surprise. Mohammed Ahmed is asking, he says, nice talk. Could you explain a little bit how you identify that you have a racemic mixture of that paraquinone based on the X-ray data? That's a very good question. Yeah, how could you tell that it was racemic from the X-ray?
So, it exists as 50 to 50 racemic mixture of the two axial chiral. We tried to draw it here or to put the crystal structure here. As you can see, the axial chiral here is going in the opposite from this one. So these two compounds are really enantiomer. And this is what we call the racemic mixture. I'm not sure if I understood the question very well, that you have difficulty seeing the difference.
Yeah, I think that's a good answer. The other answer is that thankfully we've got great collaborators who are experts in X-ray crystallography and can help us interpret those data. But that's which is a new collaboration, which we're very happy about. Okay, I don't know. No, Dr. Hamadian, is anybody else asking a question? In the chat. Okay, here's a question from the chat from, I might be saying the name wrong, but it's Hibad Dalaha. Yeah. Congratulations, Zeinab. I would like to ask the reason for choosing the medias for your experiments. Was this based on research or was it random? That's a very good question.
Thank you, Hiba, for the nice questions. We didn't really do characterization for the media, and we choose these media as kind of experience in our lab. So we use these media a lot, and they were proven to be effective in enhancing the production of these fungal secondary metabolites. So we consider this as a first step, and if it didn't work with these three media, we can work more with other media.
Okay. Are there more questions? Oh, Q&A, sorry. Okay, one more question in the Q&A here. Oh, this comes from our friends in England. So Dr. Michael Hall says, great talk, Zeinab. Some questions from the UK. Does the wavelength of light used make a difference to the production of the paraquinones? I'm going to ask you the second question too, because it pertains to the first, which was, what flavor of Cheerios gives the best production of secondary metabolites? I could tell you the flavor makes a difference, but anyway, you can ignore that question. Go to his first question.
I don't like Cheerios, and I believe it's just food for fungi. But for the other question about the wavelength, as I said, for the study, the result that I presented today, we used all the wavelength. We used the white light, but we really did another study where we studied the blue, the red, and the white, and there wasn't really significant difference between them. All of them were found to enhance the production of these compounds, so I don't think there is a difference.
Okay, thank you, Zeinab. All right, I don't know if I see any other questions. So maybe the one thing we could do around the world, if anybody would like to just sort of give her a clap and all that jazz, we're going to do that here in the room for her. And do we have anything else to do?
No.
Oh, yeah. Okay, so now we're going to hang this thing up, and the rest of us are going to ask her questions for the next 1 to 7 hours, something like that. So anyway, so we thank you all, though. Really, it's very nice to have all of you tuning in, especially Zeinab's family. I hope to meet all of you someday in Jordan, someday soon, and inshallah instead of in Charlotte. And don't worry, she'll be back there pretty soon. So thank you all very much.